Today, we're going to have a look at how large language models do analyzing gene expression data. I'm Doug Selinger, founder and CEO of Plex Research, and let's get into this example. So we're going to start off with this gene expression signature where we know where it came from this paper studying Duchenne's muscular dystrophy. So this is a gene signature from transcriptomics, actually single cell transcriptomics. Really nice study. We're going to use this as an example. So the signature from this paper, I chose the upregulated genes in a particular cell type. That's going to be our input. And so we're going to go first to ChatGPT here. So the model, I gave it the same question to all the models, and you'll see that. And here ChatGPT comes up with fibrosis. So fibrosis associated with chronic inflammation. As you go down here, it talks about why, what it found and how it came to this conclusion. Neurodegeneration is here as well, oxidative stress. It actually has a number of different hypotheses that lead to different kinds of disease associations, right? So idiopathic pulmonary fibrosis, chronic kidney disease with fibrosis, Alzheimer's disease, right? So it lays this out. Nowhere in here is Duchenne's muscular dystrophy. So really, there may be some overlap in the phenotypes of these diseases, but we are not getting the specific disease that actually generated the signature. If we go on to Claude, uh, what we see here is we get also sort of fibrosis type phenotypes, heart failure, fibrotic remodeling, supporting evidence. There's fibrosis markers, right? Cardiac specific genes, inflammatory response, all of these things. And so it's again sort of in the area of fibrosis with different reasons, trying to support its logic, but nothing here specifically about Duchenne's muscular dystrophy. If we move on to perplexity, here we end up on Alzheimer's disease and neurodegeneration as the main hypothesis. It walks through and says why it thinks that, where its reasoning is coming from. One of the genes that's modulated actually is ApoE, which is a well-known Alzheimer's gene. However, the signature as a whole, as we know, came from a DMD sample. So this is not an accurate representation of the disease state that is actually in play here, right? We kind of scroll through this, and there's really no place are we seeing anything about Duchenne's muscular dystrophy or anything really, any other muscular dystrophies or muscular diseases. So finally, if we come over to Plex, so here, Plex is able to look at this signature and immediately and with high confidence assess this is DMD, this is Duchenne muscular dystrophy. You see that coming up here. And how is it able to do this when the large language models, the other models, are not able to? The key is that it's using tools the others don't have, and it's using data that the others don't have. So Plex is using a search tool that is looking through relatively raw gene expression data. So gene expression data coming from lots of different databases and assessing the signature that we've put in, these genes that we've put in, where have we seen them before? Have they been seen to be differentially expressed in other diseases in all of these databases, right? And if so, what are these other cases where we've seen this? What is the disease state? What is the cell state? What is the biological state? So that's what it's doing, and that's something that these other models are unable to do, at least not with additional tools. And that's really that unique tool that Plex has that allows for that kind of data analysis is really what sets this apart. And you can see that here. So as it lays out the evidence, so if we scroll down a little bit here, we see DMD. So the primary evidence for DMD is that there are multiple RNA profiles that we're able to identify that have a very similar profile to the input gene list. For example, in the Plex search, the top ranked RNA profiles uh, contain, for example, this comparative profile of 13 muscle groups, which includes DMD as part of it. Also, this study specifically of DMD is ranked very highly. Again, the signature is very similar to the signature that we put in. And same with these other studies here. So there's 4 studies near the top of the list that are all either specifically DMD or very closely related, include DMD, that are all very highly ranked, matching the signature. There's also other things, and we do see fibrosis, right? Some of the other models picked up that's consistent with DMD, but isn't alone sufficient for a specific DMD conclusion. So it does pick up the fibrosis as well, picks up inflammation, picks up muscle regeneration as well. So more things linking it back to this being a disease of the muscle. And it says you could consider going, further, either more general or to other, for example, other forms of muscular dystrophy or potentially other more general things. So, but in conclusion, uh, you know, the gene expression profile here most strongly indicates DMD. So this is a pretty firm conclusion. We just saw the evidence on which it's based. It reiterates exactly what the evidence was here. Now, in order to get here, a Plex search was run. So we asked it the question, and I'll sort of go back up to the top just to show that this is the prompt. This is the identical prompt that was given to all the models. I've actually posted this prompt before, and I'll post it again along with this video. So you can try it for yourself. You can paste it into your favorite models using whatever tools you might have and see how it does. So basically, these genes were upregulated in a disease without naming the disease. And based on these results on the profiling data, what disease do you think it might be and provide evidence. And this is the gene list. So what it's doing here is taking in that gene list, resolving them, and then running a search, a Plex search. And this is really the special sauce here. The thing that we're doing that other models are not able to do, it's a tool, we're providing a tool, a unique tool. Um, this is a search that takes this list of genes and searches lots of databases for similar profiles. And if you click on this, it launches that search, which I just did a little while ago. And you can trace back the data that it's citing uh, are present in this search. So you can uh, see exactly where they came from, see the exact data that supported this, verify that in fact this is not a hallucination, that it actually did find this data, and you can identify the source of it. So for example, it said that the top result is this profiling study that includes DMD, and you can see that right here at the top. In fact, let's see, we can filter for DMD. That'll probably make this a little easier. So let's just do that. So we can filter for DMD and see that, yes, this study, DMD was part of it. As we scroll down, we see that this other study that was ranked number 5 was another DMD study. As we scroll down further, this one, which was ranked number 49. I think actually if we filter to, well, let's do Duchenne instead of DMD, because I think there's another one that we missed. So this is ranked number 5. Um, this is ranked number 14. This is ranked number 49. So there's a number of studies here that are related to the input signature. We can see here as well that, for example, this study, there are 23 genes that were in our 15 gene signature that are also modulated in this DMCD study here, right? So that's why this is coming up, so it ranks so highly. Very similar gene signature, almost half of the genes that we put in are differentially expressed in this particular DMD study, right? Actually, if we instead of Duchenne's, we filter for muscle, you see actually there's even another study. There's that first one that we saw. There's also this other study of skeletal muscle. So again, more evidence that this is muscle-based and a considerable amount of overlap here. So by leveraging all this other data, and here I'm only focusing on the RNA profile category, and you can see here in the result, actually, there's quite a bit more. We're not going to get into it in this video, but for example, we can ask, are there compounds that have protein level changes or that induce protein level changes that are similar to this disease signature, right? So potentially, compounds that could potentially reverse the signature and therefore putatively would be treatments for DMD or for related diseases, right? So can we identify other things that when perturbed modulate DMD or this DMD signature? So we can look at proteomic profiles, whether they're induced by compounds or something else. We can look at these perturb-seq profiles. So are there genes that we can knock down that will result in gene expression that is the same or maybe the opposite direction to our input signature. So there are many, many different kinds of data that we can link to that beyond just identifying the disease state could give us options for or ideas about how to modulate that disease state. Or things, whether genetic perturbations or pharmacological perturbations, that could potentially reverse or mimic the disease state, the gene signature that we put in, right? This also having such a wide array of data sets, even within the category, there's many different kinds of model systems and kinds of studies, right? So having this variety lets us more deeply characterize exactly what we're looking at. And we see that pretty convincingly already that this result leads us very specifically to DMD, right? Um, it can also lead us to potential modulators, again, whether pharmacologic kind of drug modulators or genetic modulators or any other kind of modulators. It can lead us based on the kinds of things it's matching to other maybe unexpected connections that give us insight into the underlying molecular mechanism, the underlying disease mechanism that's in play, potentially with, again, with a very high level of specificity. So rather than being stuck in, maybe the standard analysis or something like we saw with the other large language models where they said, oh, it's something about fibrosis, something in this general area. Here we can link far more specifically to a very specific disease state, a very specific disease model. It may be context dependent. It may be that you only see this match to a particular cell system, model system disease, maybe only a particular subtype of the disease, maybe only at a certain time. So a certain kinetics could be involved, could be a dose dependency, it could be a genetic dependency, could be all kinds of things. And those sorts of details and specificity can be surfaced in this kind of analysis by looking, casting a very wide net across many data types and many, many kinds of studies and identifying where there are matches. And again, the fact that we can very specifically identify DMD is just kind of the tip of the iceberg. It's just the indicator that we can, in fact, achieve a high level of specificity while maintaining transparency about how we made that connection, exactly what we're connecting to, what this might mean with all the details, because each of these studies, there's tremendous details, right? We can link for any of these results, we can link back to the original studies and see exactly what was done and understand better how to interpret that precisely. Um, so it's really a pretty compelling result. I'll also point out that this is all here, right? So it went off and ran that search. It's actually a multi-agent search happening here. There's this guide agent that's deciding what to do, what searches to run, and it's handing off that search to this analyst agent in green here that runs the search, looks at the result, and feeds back its conclusions. So as we scroll down, we can kind of see its work. We can see the analyst's work. Um, you can see it's sort of feeding back, looking at the result and feeding back what it's seeing. I won't go through all of that. Here is what it ultimately feeds back to the guide, and you can see some of these results kind of being fed back. And as we go down further, it's running additional searches and finding out more information, right, about the results that it saw, and then it makes this conclusion that we just walked through. Now, a really nice thing here, beyond just this conclusion, which of course we can export, so we can capture this, both the text and also JSON, so we can archive this exact result and reconstitute this session exactly where it is, and potentially run different parts of it, have it start from somewhere else or have it continue, right? Because we can have a follow-up question here. So we can archive this and capture it. A really nice feature as well is that we can create a slide deck. So with one click here, it takes about 30 seconds, and a slide deck comes out that summarizes what was found. So here is what that looks like. This is one. This is the example we're looking at here. Uh, Took that result, and this is what it produced. And I'll just bring this up as a slide deck here. And so this is entirely unaltered. This is just click that button, and this is the slide deck that comes out. So including the title and all of that, muscular dystrophy gene expression analysis. As we go through this, the executive summary. So what tells you what it did? It looked at profiling of 50 upregulated genes. That indicated that this is a DMD disease state that it's looking at. It tells you here's the evidence behind it, that there's especially these multiple RNA profiling studies. and sort of summarizes what it found. Again, summarizes the approach, took those 50 upregulated genes, looked for patterns across many categories, right? So we focus on results in RNA profiles, but as I mentioned, this casts a very broad net. So if this signature was seen or something similar in some kind of proteomic response, so not just RNA, but protein, or in perturb-seq result, which is also a kind of RNA data, genes knocked down, and RNA expression analysis after those gene knockdowns. So if there was some gene that when you knocked it down gave a similar signature, that would come up here as well. So that can actually give you insight into, what are the genes? Are there other genes that are involved in DMD, maybe that were not appreciated, right? That we can discover that through the data that knockdown of that gene gives a similar profile to our disease signature, right? So this is a way to start off with the disease signature, uh, benchmark it effectively because we see that we get very strong connections to other independent studies of DMD. So we know that this disease signature is generalizable and robust. And now we can look at potentially other categories or even other things within the RNA profiles. And I just sort of breezed through by some of them you may have noticed. There could be connections to other RNA profiles that are perhaps unexpected that could represent new discoveries. So we can discover potentially other genes or maybe pharmacological perturbations or other diseases even that might have thought to be different, but maybe share an underlying mechanism. So we can come in with a disease state and potentially discover new genes that are involved with this disease or that could modulate this disease, new pharmacological modulations, so drug treatments, repurposing applications, or other diseases that are maybe related to our disease of interest that could be affect our strategy for disease indication expansion. So we have a drug for one disease, and now we discover here's another disease or two that share an underlying mechanism, and therefore patients with that disease may be responsive to my therapy to our original disease, right? So disease indication discovery for a therapeutic, disease indication expansion, drug repurposing, all these applications just emerge very naturally from this sort of approach. And we could even ask the LLM to address that specifically so that the slide deck we get out actually addresses that, talks to that. And of course, we can ask it to analyze the Plex results with that in mind, right? So looking specifically rather than, as we did here, asking it, tell me what disease state this is, we can ask it, identify other diseases that we can expand to, that if we're addressing this disease, what other diseases are related, right? So summarizes the approach, tells you this is why DMD is the answer in this case, or is the strongest result. And in this case, we know the source of the gene signature, so we know this is the correct result. Uh, so it tells us just what we saw a moment ago. In the RNA profiles category, the very first study is a DMD relevant study, even though it does cover other diseases. Then ranked 5, 14, 49 were studies that were very specifically looking at DMD. So this was really what made this case the strongest. This is again data that is not being used or as far as we know by the large language models, and even if it is, there's this lack of transparency where we wouldn't know if it did come up with DMD. Let's say, Claude or Perplexity or whatever it is did come up with DMD. It might be difficult to understand why, right? Whereas here, because this is from tool use, we can launch that search. We can look at the results of the search and say, the reason that this signature looks like DMD, or we think it's DMD, is because of the similarity to the following specific studies with all of the details and the links back to the original studies, right? So it's that transparency, both of the approach, right? So it told us what it did, and the underlying data. We can evaluate these studies, the amount of overlap, the strength of the connection to DMD, certainly within this category, potentially across other categories of data. So there's really many ways to come at it. And then in addition to those specific studies, which is really what gave us the specificity, there's also these more general aspects which are consistent with DMD. They're not alone sufficient to make a specific diagnosis of DMD, but they support a DMD disease state. And so it talks about basically fibrotic response, and this is something that the other models picked up, but couldn't link to such a specific disease as we did. Okay, so walks through just whatever else it thought was notable, and talks about, what you could do with this, the novelty of it, uh, drugability, if you were going to think about potential ways to treat DMD, new approaches to DMD. This would this sort of approach where you understand the underlying mechanism can be a first step, right? An important step in identifying new ways to treat the disease or to diagnose the disease or to understand the different subtypes or who will respond to a therapy and who won't. And many, many things come from this. So this can be a part of the drugability and kind of the therapeutic approach strategy. Commercializability, these are things we sort of have it look at as standard practice when it generates these slide decks. And of course, we can continue to dig into this with Plex. There's always room. You always need to go back to the lab at some point and do the next experiment. But there's a lot you can do here just with this sort of approach, because we can look at these other studies that were identified as being similar. Gene lists from those studies can be used as input to Plex. Compounds that are maybe linked, that we've been able to link to this disease state, can be analyzed in Plex, including with their chemical structure. And that will be the topic of another video. So we can infer potential targets and mechanism based on the chemical structure of compounds. So there's a lot of things that can be done with this, and this all summarized here. So, so what I hope I've shown you is that there's really, with the right tools, we can do a whole lot more than we realize. And in fact, there is a tremendous amount of data in the biomedical space, even just publicly available data. There is a huge amount of it. And this kind of approach that is taking large language models, and we haven't trained our own model here. So this is an off-the-shelf foundation model, but what we've done is provide it with the right tools. So the tool being Plex search. So there's of course internet searches. This is a specialized search based on focal graph technology that we developed that allows us to run searches, very highly concise, informative, and very comprehensive searches of very wide-ranging, diverse, complex biomedical data. So gene expression data, which could be RNA and protein or genomics data, chemical structure data. Again, that will be at the topic of another video. We can mine all of these different kinds of data and come back with very, very concise, but traceable, transparent, supporting data uh, that enables, when paired with a large language model like we've done here, enables these studies that can actually make new discoveries, right? Can connect to something that has never been connected to before and give us an insight that maybe has never been published before. And again, we'll get to that in more detail in future videos. Can surface connections, novel connections, novel targets, novel mechanisms in biology, because it's using relatively raw data. So this minimally processed data about what genes are going up and down, and what proteins are touching, and what genes are associated with something, and also chemical structures and their bioactivities. So using that minimally processed data, of which there is an absolute abundance, we can make inferences and make connections, and as you see here, that can support new discoveries. So not just rediscovering what is in the literature, but making new connections, identifying new mechanisms, new targets that can actually really change the game in drug discovery and in medicine. That's really the goal of this approach and what we're building here. So again, there'll be more examples. This is really, I tried to start off with a simple example here. I hope this is a simple example. And we'll get into a lot of other use cases in the future. This is one that I encourage you to play around with yourself. So I'll provide, as I've done actually in the past, the exact prompt here, so you can try it out with other models, with other approaches. Uh, and there's really a lot more to come here, a lot more things you can do. that I think will finally start to realize the promise, the excitement around AI, the hype around AI, and the open question of whether it can really contribute to actual advances, to true advances in drug discovery and medicine. I think it certainly can, if if done in the right way. And I think and we're showing that using this approach, we can actually make advances. We can actually make new discoveries. We can actually move the needle. And this is just one demonstration, that one example of that. There'll be more to come. So maybe I'll wrap it up there. I really do appreciate your interest. If you'd like to learn more, as I said, I'm planning to make some more videos covering other use cases. There are a lot of different use cases we can address. Please visit our website. There's a lot more information there, plexresearch.com. I'll have the link in the information. Also, check out our preprint, our bioRxiv preprint, which describes focal graft technology, how that works, how we develop that, the various applications of it, including this example. So this example is mentioned in the preprint. I've gone through it here in more detail than it is in the preprint, but we do, as far as at least the results. But it is discussed in the preprint. So again, plexresearch.com. Have a look. Let me know in the comments what you think. Is this moving the needle? Is this, have you tried this yourself with your models or other approaches? How did it go? Let me know in the comments. And thank you again, and look forward to seeing you in the next video.